Thank you. So it's great to be here, and today I will be telling you a bit about blueprint genetics and what we do at the field of clinical genetics. And more importantly, I'm going to talk to you about rare diseases. So the rare diseases are devastating diseases that affect over 350 million people around the globe. These are really devastating and life-changing diseases. So in many times they dictate the lives of the patients if they are in constant pain, if they can have children, if they can marry, if there are treatments available for them. So understanding and getting answers to the root cause of these diseases is extremely important to these patients. But getting to those answers is not easy. When you sequence a person's genome, you get over 100 billion data points, and out of those 100 billion data points, you get over 5 million personal DNA variants that make up your personal genetic profile. And usually, out of those over 5 million variants, there is only one that actually causes the disease. And so finding that single disease-causing variant is actually a quite big task. So how do we actually do this at the Blueprint Genetics? We are full end-to-end clinical genetic diagnostic company. So we take in patient samples, we do DNA sequencing, we do a lot of data crunching, and then we interpret the data and send back a clinical statement. We do believe that a lot of this work is, if not easy or trivial, it's still manageable, especially the first part, the first parts, the DNA sequencing and so on. But especially the interpretation of the data is one of the major hurdles. We right now have around 30 clinical geneticists and medical doctors plus 15 medical specialists who work interpreting the data and trying to understand what are the disease-causing variants in that data. So, and to give you a bit of background what we are talking about and what amounts and types of data we are talking about, I'll tell you a bit about my own history in this field. So I started working with genetics in 2002. Around that time, there was a big project called Human Genome Project that was just finished, a massive project, took over 20 years, cost over $3 billion, and the goal of that project was to sequence a single human genome that the whole humanity could use. So, quite expensive. In 2006, I went as a visiting scholar for a couple of years to visit Broad Institute of MIT and Harvard, one of the world's leading sequencing centers and technology developers. At that time, people there were talking about promise of sequencing a human genome under $1,000. And I was thinking that that's a crazy talk, because that year, the second human genome was sequenced, the genome of Dr. James Watson, Nobel Prize winner who got the Nobel from discovering the double helix structure of DNA. Sequencing that genome cost $100 million. But next year, another genome came that cost $10 million to sequence. Next year, $1 million. And so in around 2015, we actually broke the magical barrier or mark of a $1,000 genome. And right now, sequencing of a single human genome costs some hundreds of dollars. So it won't be long when we break the $100 genome barrier. So just during my lifetime, or not even my lifetime, but my professional career working in genetics, we've gone from over $3 billion to something like $300. So this has really caused a paradigm shift in this whole field. And this is also aligns nicely with the history of Blueprint Genetics. As mentioned, Blueprint Genetics was founded in 2012, and actually around the 2015, our company actually started to see also exponential growth. So we've been diagnosing tens of thousands of patients during our time, and we are really growing rapidly, doing this in a really growing pace. So each time we process a batch of patient samples, we create around 5 to 10 terabytes of data. And I noticed that there is a mistake in this slide. It says 200 computing hours. It should be 2000. So this is something that we do several times a week. We press a button on our sequencing instrument and create these up to 10 terabytes of data and crunch it in a cloud computing environment using over 2000 computing hours. When you talk to a lot of companies who say that they work with big data, these are usually the amounts of data that they have accumulated during the whole lifespan of the company. For this is a single run of one of our experimental equipments. But so this is routine. We are quite good at handling these amounts of data. This is easy to us, but the real challenge comes from the fact that the number of sequenced genomes worldwide doubles every year. It's estimated that next year over 30 million genomes will be sequenced worldwide, and around 7 million of these will be for clinical testing. And currently, this is just an estimate, but if you take all the clinical geneticists and medical doctors working on interpreting the data, all of the companies, all the geneticists working in hospitals and so on, we estimate that around 300,000 genomes are clinically interpreted per year, and next year that number should be 7 million. It's quite hard to multiply the number of all of these experts worldwide. So, what's the solution? At least we believe that we need to empower the geneticists by providing augmented intelligence for interpreting the genomic data. We really want to automate as much as possible of this process, increase speed, but also increase quality. So provide results that cannot be accomplished just by human experts, no matter how good they are. Towards this, we've been developing our own software tools, as well as our own machine learning based methods. And in practice, this means that certain bottlenecks that you can help with the help of machine learning and augmented intelligence is highlighting relevant information, meaning that from all of these massive amounts of data, which is further enriched with data from different databases and so on, the clinician or the geneticist working with the data only needs to look at the relevant data that's automatically brought to them. Another feature is to suggest the right literature, because every time a geneticist is making a decision that this is the disease-causing variant, this is the thing that has caused the disease that the patient has, it needs to be rooted in existing scientific literature or case reports or something that shows why just that variant is the disease-causing one. And so finding the relevant literature out of all of the hundreds of millions of scientific publications out there takes quite a lot of time. So finding the literature is a key point here. Another one is variant classification. So you have 5 million variants in a patient, and you want to classify them as benign, so non-disease causing, and as pathogenic, as so disease causing. And usually there's only that one single disease causing variant out there. I will talk a bit more about this later. Another thing is collective intelligence. So reusing the information when you've encountered a similar patient case or similar variants in the past. So not needing to do or redo the same work, but utilize all of the existing information. And another thing is the statement generation. So the end result of our service is a clinical statement. And the clinicians who read those, as you probably know, are busy people. Often they have only a couple of minutes to read through the statements before meeting the patients. And so in that piece of paper, a couple of pages or whatever, you need to describe what was done and how, what's the conclusions and what's the important parts of those billions of points of data, and what's the relevant thing there, and produce this in a natural language that takes into account also the medical history of the patient. So this is not an easy task to automate either. But so, talking a bit more about the variant classification, luckily machine learning is actually really good for classification of things. So the idea is that we have 5 million DNA variants, and we need to pinpoint that one disease causing one. So we've been experimenting with this for quite a long time, creating prototypes and testing this with actual patient data. And we've learned that this actually works quite well. So when you create the machine learning classifiers that use information from public or private variant and gene databases, there are a lot of tools out there that do this already. But to get to a personalized precision medicine level, you need to personalize the model with the patient-specific information, such as patient characteristics like sex and population, genetic population where that patient comes from, phenotypes such as symptoms or disease classifications, family genotypes if they exist, and sample-specific information like sequencing quality for that one specific patient sample. So we do believe that the winning formula for this, you cannot totally automate this. And on the other hand, there's too much data and information for humans to handle right now. So we believe that you need to have the best people in the world and provide them with the best tools. And this is the thesis that we are at Blueprint that we are working on right now. So what do you actually need to get there? You need solid real patient data for training and validation of your models and algorithms. So it's really important to be handling actual patient data on a daily basis and gather a massive collection of real patient data so that you can actually even create these software tools. You need to understand the genetics and patients. So I mainly lead the software development teams at Blueprint Genetics, and our hardest supporters and critics are our co-workers, the geneticists that work next door. They work almost day and night interpreting these patient cases, trying to solve what's causing the disease on the 3-month-old baby, or how do we save the life of the patient suffering from some really drastic disease. And they want that these tools are top-notch and they serve the real customers who are the patients. As a clinical testing company, we are of course accustomed to external and third-party validations. As scientists, we embrace clinical trials and so on. We've noticed that this is not always true with all of the companies working with the AI and machine learning. People are quite scared about someone else evaluating how these things work. But I think that there is a huge need to expose yourself to this kind of external validations as well. And I cannot emphasize enough the importance of these cross-functional teams, meaning you need software developers, data scientists, but also geneticists, medical doctors, lab researchers, and so on, to really understand what are the needs and possibilities of these different methods. And now that I've been speaking mostly about the possibilities and opportunities out there, I always want to spend some time talking about the risks and challenges that actually need to be solved. So these methods, first of all, they are often limited to certain types of DNA variations, for example, protein altering variants, which are the most common and usually the most likely disease-causing variants. But there are a lot of different types of variants. So there are long deletions of DNA, there are replications of DNA, big structural variations in the DNA, and these methods don't usually work very well on those. There is selective bias, as with any AI or machine learning methods, basically meaning that the data that you use to train these models is usually somehow biased. When we are talking about genetics, most of the genetics data in the world is generated from white European populations. The most sequenced population in the world is Icelandic people. Second one is Finnish people. So a lot of these methods are trained on data that works really well for a population like what we have here right now, but they don't work so well from people from Southern America or Asia or a lot of other places in the world. Same goes, of course, with the different diseases. There are a lot of incentives why certain diseases or types of diseases are more researched and studied than others. And this will always create bias in the training data. There's the interpretation of black box results. So how well do you need to understand how a computer generates some kind of a diagnosis? Are you just happy if it's usually the right one, but do you need to back it up and explain how the computer came to that conclusion? There's the regulatory accountability of AI and machine learning models. When we are working with clinical testing, there is a lot of regulation. We need to be able to replicate our results, usually years after those were produced. So go back in time and say that, why did we do that type of diagnosis 3 years ago? Even though when we are accumulating more and more training data all the time, we need to be able to reproduce and explain step by step how we came to a certain conclusion and how our model was working back in the day. And one really interesting point is data recycling. In the field of genetics, there are a couple of public databases where there is a lot of genetic data available for the researchers and companies. The data has been generated by public funding and is an enormous resource for anyone wanting to develop these tools and methods. But when you take that data from that public repository and use that data to create a tool, and then you use that tool to create more data that you put in the repository, that you then use to create another tool, it creates this kind of cycle where the initial data will bias all of the tools, methods, and all of the data that's generated in the database in the future. So, and then also to mention one additional challenge. I'm wrapping up, but I usually want to show this one tweet that went viral a couple of years ago, saying when you are fundraising, it's AI, when you are hiring, it's machine learning, when you are implementing, it's linear regression. So every time you go to a conference like this or any else nowadays, be critical, think about what you are being told, ask questions, and keep it real. So that's all I had for you guys today. Thank you.